We've got the cranial cavity, and then what organ is found within this cavity right here? The spinal cord. This is the dorsal body cavity. What's another term for dorsal? Posterior. Good. What's another term for anterior? Ventral. So this is the ventral body cavity. What organ is found here within the cranial cavity? The brain. The brain and the spinal cord, these are parts of the central nervous system. Look at the ventral body cavity. We can further divide it. The structure that's going to separate them, that's what this structure is right here. This is what's known as the diaphragm. So this structure separates these two different cavities here. And what is this cavity here at the top? Which one is that? Thoracic. This one here at the bottom. So we have as a whole, it's known as the abdominopelvic cavity. So this is the pelvic cavity, and then this is the abdominal cavity itself. What organs are going to be found here within the abdomen? The liver, the gallbladder, the spleen, the intestines. Found within the pelvic cavity, you've got the urinary bladder, you've got the reproductive organs found within this body cavity. As a whole, this is the mediastinum, and then at the top, that would be the superior mediastinum. This is where the heart is found, and it's found within the pericardial cavity. Here on the side, this is the pleural cavity, where the lungs are found. The relationship between the serous membranes. If I put my fist into a balloon, this balloon is going to be filled with some water. That inner lining that's directly surrounding my fist, that is going to be the visceral layer. And then the part where it folds on itself, so see how it's folding on itself right there? That is the parietal layer. With the visceral serous membrane, this is what's going to be in direct contact with the organ itself. So the specific example that they give here that they show is the pericardial serous membrane right here. The fluid that's going to be found in between the visceral and the parietal layer, that is going to be our serous fluid. So that is our serous fluid, and the benefit of us having this fluid in between these two different layers, it's going to prevent some of that friction. Quadrants, and you also have regions. We're specifically talking about the abdominal pelvic region. We can divide it up into right, left, so right, left, and then the upper quadrant, and then the lower quadrant. The way that I'll ask you guys some of these different questions, I'll say, what organs are found within the right upper quadrant? Or all of the organs are found within the right upper quadrant except what? So remember, you're referring to the observer. You're looking at their right and their left. My liver is with an R, liver, so it's on your right side. The majority of it is found on the right side, on the right upper quadrant. The gallbladder that ends with an R, that's also going to be on the right side. The stomach is just going to be on the opposite side within the left upper quadrant. What structure is going to help distinguish between the upper versus the lower quadrant? What was that?
The large intestine?
Yes, the large intestine. The large intestine or the colon. So specifically, this is known as the transverse colon right here. So once you get past the transverse colon, that's whenever you get into the lower quadrant. What structure is going to be separate the thoracic and abdominal pelvic? Diaphragm. Spleen is going to be found right here on the left upper quadrant. It's a little bit harder to see. What is the part that lines the internal body cavity walls? Which serosa? Which layer? Visceral or parietal? Everyone at once. Parietal. What's in direct contact with the organ? Visceral. So they are named for their specific cavity and organs that they are associated with. Pericardium, you know that the heart is going to be found there. For the pleura, you know that that is going to be the lungs. The peritoneum, that is within the abdominopelvic cavity. So this is the ascending colon, and then this is the first part of the large intestine known as the cecum. So you've got the cecum, and then this is the appendix, and then it goes upwards this way, and then it travels, so that's the transverse, we talked about that, and then we looked at, this is the descending colon, sigmoid, until it becomes the rectum here towards the very end. So once again, be familiar with these quadrants as well as these different regions here. What is matter? It's anything that occupies space and has mass. So when we refer to mass, we're specifically referring to the matter only. We look at your mass on Earth. It's 50 kilograms. And then if you go to the moon, does your mass change? No, it doesn't. It stays the same, but why does your weight drop? It goes from 490 newtons to 80 newtons. Well, it's due to the lower force of gravity. On Earth, it's 9.8 meters per second squared. On the moon, it's 1.6 meters per second. Weight is going to consider gravity. States of matter, and these are the definition of them, but I wanted to ask you guys, so what are some human body examples? Give me some examples of a solid. Bones, that's a good one. Organs, yeah. Organs, yeah, like for instance, like the stomach. So obviously the fluid that's on the inside of the stomach, that would be considered a liquid. Bones, also the teeth, the liquids. You think about what circulates in your blood plasma, gastric juice, that would be a liquid. As well as think about interstitial fluid. That's another example. Gases. Think about your respiratory gases. You're inhaling oxygen, exhaling CO2, as well as gases in the colon. That's another example after fermentation. Energy. So how do we define energy? We're going to measure its effect on matter. What is it able to do? It's able to do work or put matter into motion. It does not have mass, and it does not take up space. We can use energy that's going to be stored in order to put things into motion. Potential energy, this is going to be an inactive form. It's not in action yet. It is stored. This is stored energy. As opposed to kinetic energy, this is going to be energy in motion or action. Either is fine. An example, a Humpty Dumpty, or if you think about a hill here, if we have some type of object, whether that's a ball, we have it at a particular height on top of this hill. Once it starts to roll down this hill, what's going to affect it here? Gravity, yes, so gravity. And then we also consider the actual mass of the object itself. We are converting this potential energy that's in this ball, and then you roll it down the hill, it gets converted to kinetic energy as it starts to go down. So then we look at the equations here. So potential energy is mass times gravity times height. So all that is going to, so the higher it is, that hill is, the more potential energy is going to be stored. And then when we compare that with kinetic energy, however, we compare that mass and then the velocity at which it's going to travel as it's rolling down that hill. Describe the major forms of energy. One is chemical. So we'll look at this molecule here. This is going to be ATP. This mechanism here is called ATP hydrolysis. So we store this energy here at these phosphate groups right here. What are these called whenever they're linked together? Bonds. So that is where the energy is going to be stored here. It's stored in the bonds of the chemical substance. And the example that we looked at was ATP. Electrical energy. We can compare household items here compared to a biological example here. What's moving here is the electrons. We move the electrons here, and then when we look at the plasma membrane, the sodium is what enters into the cell, and so it changes the membrane polarization. It becomes more positive. What is the common theme between these two? We have movement of these electrons and then movement of the different sodium, these positively charged things. What is the common theme here between the two? What are they? They're charged. So we have movement of charged particles. Electromagnetic energy, this is what's going to travel in the waves. So you look at the wavelength right here. This is going to have a longer wavelength We look at, until you get to this point here, later on you start to get into the gamma rays, but very short here. So electromagnetic energy is going to travel in waves. Mechanical energy, which this is going to be just direct movement of matter. So whether you're riding a bicycle, hitting a nail with a hammer, that is going to be direct movement of matter. Converting these different energy forms here. Energy that's in a light bulb, the movement of these charged particles, that is the electrical. When these photons get released here, what type of energy is that? Electromagnetic, more specifically, visible light, this part of electromagnetic energy. So we're converting electrical into electromagnetic energy. If you touch that light bulb, it's going to be hot because some of that energy is going to be lost in the form of heat. At a car, why do you gas up? What's found within the gasoline? You've got all of these different chemical bonds, all of these hydrocarbons that are going to get oxidized. What this enables, the motor, the engine of the car, we're using all of that chemical energy into what? Mechanical. So on the inside of the engine, there's a spark plug, and some of that fuel gets injected into the engine. Once it lights it, we have expansion that causes the movement of the piston. It's connected to the crankshaft, which is going to help to turn the tires on the car. So we're converting that chemical energy into mechanical energy. Elements cannot be further broken apart into simpler substances. All elements are made of atoms. Do all elements have the same number of subatomic particles, protons, electrons, and neutrons? No, there's going to be variations in them. Due to these variations, that's what's going to give them their unique physical and chemical properties. So just distinguishing between the two physical properties, things can be measured. Density, solubility, melting point, all these different things, and they can also be detected by our senses. When we compare that with chemical properties, we're looking at their actual bonding behavior, how they actually react, these different elements. There's 4 elements in the human body that are going to make up 96% of our body mass. And so I have it listed in order. So oxygen, carbon, hydrogen, nitrogen. Where these elements are going to be found here, what they're involved with, it's a component of organic and inorganic. When we say organic versus inorganic, on the thing where we say organic, we refer to carbon. But you cannot exclude, you have to have hydrogen in there as well when we say that it's organic. And as a gas, it's going to be required for, we talked about this earlier, ATP synthesis. Inhale it through the respiratory system, transport it using the cardiovascular system in order to get it to our cells. Carbon is going to be found in our macros, right? The foods that we consume, carbs, fats, proteins, nucleic acids. What does hydrogen influence as a free proton when it loses an electron? It influences our pH. Influences the pH or the negative log of the proton concentration. So we'll talk more about that as we move on through chapter 2. Nitrogen, this is a component of proteins and nucleic acids. We are starting at the atomic level, guys. And I know it seems kind of like, oh my gosh, I already know this is review, but it's going to build on itself. I have to make sure that you guys are aware when we start talking about the structure of an atom, because all of that information is going to start to build on itself when we start talking about all these different components, and then how it's going to, as we further and further move on, start talking about solutions, and then we're going to start talking about how the different types of bonds, the ionic bonds and polar bonds, a form of a covalent bond. All this information is going to build on itself. An atom is the fundamental unit of matter, and it forms elements, ions, compounds, biomolecules. Ions, these are things that are going to be charged. The 3 subatomic particles that make up an atom, here within the nucleus, you've got your protons, right, positively charged, and then you have these neutrons. There's no charge or neutral here. And then the electrons are going to be circulating found here on the outside of the nucleus. Right? That positively charged thing and then the negatively charged thing is the opposite charge. So it's keeping that electron, keeping it close. The mass, we know that the the neutrons, and then as well as the protons, they have mass. But what about these electrons? Do they have mass? They're very, very tiny and they're negatively charged. So they have 0 atomic mass units. They have virtually no weight. So we say that something is electrically neutral if we have the same number of protons and the same number of electrons. And then you look at the location, protons and neutrons, that's found within the nucleus. Compare elements with atoms. Elements are composed of atoms. Atoms are composed of small particles, protons, neutrons, and electrons. We just talked about that. Going to vary in the number of subatomic particles. So we'll take a look at that here in a little bit. So chemists devise models of how subatomic particles they are put together. So one you have is the planetary model, and then the other one is the orbital model. The planetary model, so shown here, it's like a simplified way to discuss. We'll start talking about like the different isotopes and so on, but that's this model over here. Orbitals, think back from chemistry, so s orbital, p orbital, sp3 hybridization. Right? Those are all the things, a probable region of where an electron is going to be found. Identifying elements, so different numbers of subatomic particles. So let's look at an example here. Hydrogen has how many protons? It has 1 proton, and it has 1 electron. Helium has 2 protons as well as 2 electrons, and then 2 neutrons here within the nucleus. Lithium, that's going to have 3 protons, and it's going to have 3 electrons. But what differs here? Differs in the number of neutrons. So keep in mind here, remember, they are neutral here. The number of protons and electrons match each other. Atomic number versus mass number. So I'm going to start with the elements or the different definitions within the periodic table. This number that's at the top, does anyone know what this is called right here? It's the atomic number. What does this tell us? The number of protons. This variable that's found here at the bottom, what is this? It's the atomic weight. And so we'll look at the difference between atomic weight versus atomic mass. So the atomic number, we already said that. This is the number of protons. This is unitless. There is no unit, we're just saying the number of protons. When we compare that with the mass number is different in that it considers the number of protons as well as what? Neutrons. This is the way that they write it out here. Mass number is the number of protons and neutrons. And so this is going to have a key thing here when we start talking about isotopes. So we'll look at that here in a sec. So the atomic number, this is the number of protons. The atomic mass is not the number of protons, it's going to be the mass. It says it in the name, the atomic mass. The mass of protons and neutrons, and so this one we does have a unit. It's not unitless. It's one AMU or 1 atomic mass unit. This considers 1 single type of isotope. When we talk about an isotope, what differs in isotopes? So it has the same number of protons, differs in the number of neutrons. When we think about these different terms, the atomic numbers, are they going to be the same or are they going to be different for these different isotopes? Yes, they're going to be the same. But what's going to be different? The mass numbers, right? They're going to be different because that considers the number of neutrons. These are all different isotopes of hydrogen. This one does not have a neutron, this one does, and then this one has 2 of them. And once again, atomic mass, we're just considering 1 single isotope. However, what you see on the periodic table, the atomic weight, this considers all of the natural isotopes. There can be, like we said here with hydrogen, there's 3 different variations of it. It's like atomic mass in that it considers the mass of the protons and neutrons, except it considers the average mass of all isotopes. We'll look at an example. So it's the weighted average of all naturally occurring isotopes, and it's found in the periodic table. So what I'm going to do, I'm going to highlight this, guys. Make sure that you're keeping these things straight as far as what's on the periodic table. How are these different how are these terms similar and different? So let's look at the example here of boron 11 and boron 10. 80% in nature is going to be boron 11, and then the other 20% is for boron 10. So what you do, you multiply 11 times 0.8. Number that we'll get is 8.8. So then we consider the other 20%. We multiply 10 times 0.20, and that's going to give us 2. So the atomic weight for boron is going to be 10.8. And what is my unit here? AMU, atomic mass units. Any questions regarding this? Yes.
Wait, you just did it with the 80%? I don't understand that.
Earlier I said that we're considering all of the natural isotopes. With hydrogen, there's 3 of them. With boron, there's only 2 types. It's boron 11 and boron 10. So the only thing that differs is the number of neutrons. So it would be 5 compared to 6 neutrons. We're just multiplying because 80% of boron 11 in the world has a mass of 11. So we just multiply that to get 8.8. And then for boron 10, we just multiply the rest of it, right? 20% of it is going to be boron 10. So then you just multiply and then add these 2 variables together. Radioisotope, how are they useful from the medical standpoint? So radioisotopes, they can be used to detect cancer or damaged tissue. It basically lights up this tissue. We have that radioactive compound that's linked to that has our target molecule here. And this targeting molecule recognizes this receptor down on this cancer cell. Once it binds to this type of tissue, having this radioactive compound here, we can light up that tissue whenever we use a PET scan. So we can use a PET scan. So we can detect where that cancer is. And so we'll look at the 3 different types of radioactive decay. And the first one is the alpha decay. So this is the ejection of the helium nucleus. So it's He4/2. What does this number indicate? The number of protons, and this is the protons and neutrons. For beta decay, we have the ejection of an electron. And then gamma emission, this is just emission of photons. Or we could say electromagnetic radiation. Mixtures. So we look at molecules, it's more of a general term. We're saying that 2 or more atoms are going to be bonded together, and if it's 2 or more of the same kind, the way we term it, it's a molecule of oxygen, okay, or a molecule of hydrogen, right? If that was H2. These compounds, they're going to contain more than one type. So that's the difference between the 2. So this is, they're the same, and then here we're going to have more than one type. So we look at some examples, carbon dioxide, water, these would be examples of compounds. So we look at this hydrogen gas, is that a molecule or a compound? Molecule, they're both the same. The same atom is going to be bonded together. If we look at methane over here, we've got more than one type, carbon, and then 4 of these hydrogen atoms. Same thing with water here. So this is going to be a compound. Before we start talking about some of the specific mixtures, let's get some of the terminology here. This solution here, so we pour some sugar into this water. As we start to mix it up, the water starts to dissolve that sugar. How would we describe this solution? Would we say it's homogeneous or heterogeneous? Homogeneous. So this is a homogeneous mixture. So when we say that it's homogeneous, there is an even distribution of the solute. The sugar is the solute and the water is the solvent. and we add a solid with a solvent and we form a solution. When we mix sand with water, what would this be an example of? Heterogeneous. Is there an even distribution of the solid inside that water? No. What's formed here at the bottom? Sediment. This is the term that we use to describe it. So there is sediment that's going to form here at the bottom. Solutions. We've already described what this is. It is a homogeneous mixture. So it can be solids, liquids, gases, solid particle size. So it's very small. We cannot see these individual grains of the sugar there. This small, so we talked about that, and what it's going to do, it prevents filtration. In order to separate some of these particles out, we have to use distillation. Examples like the salt water. What you do, you have this thermometer set up, and you can separate the components based off of their boiling point. So you just add a heat source there where you boil it, and then based off its boiling point, you'll get the formation of our distilled water, so we can separate the salt from the water. Do solid particles settle out? Do we have any sediment that's going to form at the bottom here of our sugar solution? No. So sediment does not form. Do solid particles scatter light? If we were to shine a light here into this solution, is it going to change the pathway of light? No, it's not. So there is no change in the pathway of light. So solute is the smaller component, and the solvent, that is the larger component. And what does it do? It dissolves that solute. We add the solvent and the solute, and we form a solution. So what I want you to do, tell me what is the amount of glucose I need in order to make a one molar solution of glucose. So this is all the information that you need. You need this, and you need this. So the first thing we do is we look at the molar mass, and we're using this molecular formula, so all we're going to do is multiply. For carbon, there are 12 grams in 1 mole. And what do we multiply that times? 6. Times 6. And we'll get 72 grams. Hydrogen here. We've got 1 gram per mole. We multiply that times 12 grams, and then we have oxygen here, which is 16 grams per mole. We multiply that times 6, and we'll get 96 grams. We need 180 grams of glucose into how much water to get a 1 molar solution? We add that to 1,000 milliliters, or 1 liter of distilled water, in order to get our 1 molar glucose solution. Molarity. Molarity is expressed as moles per liter. 1 mole is going to be equal to the compound's molecular weight. 1 mole of any compound is going to be equal to Avogadro's number, which is what? 6.022 times 10 to the 23rd. So that is a lot. That is a lot. That is a very big number. How many grams of water is 1 mole? 18. You got 16 grams per mole for oxygen, and then you just add 2. Methane here, what would the answer be for this one? 16. Right, you just take 12 plus 4 to get us 16. That's 1 mole. But then also, another one is colloids. So that's what this is over here. So colloids, or emulsions. And this is going to be a heterogeneous mixture. These are going to be, the particle size are going to be larger compared to what we saw within solutions. And do these particles settle out? Is there any sediment that's formed here at the bottom? Looking at this example, no. We have no sediment formation here. Do solid particles scatter light? So when we shine a light here through that colloid, is it going to change the pathway of light? Yes, it is going to. Some of these different examples, so a relevant biological example is going to be within the cell, you have a bunch of these different proteins, enzymes and so on. And so at the cytosol or the cytoplasm, that would be an example of a colloid. A non-biological example here is Jello here. It changes from a liquid to a solid. From a liquid to a solid whenever you cool it in the refrigerator. Suspensions. This type of mixture, this would be an example of a suspension. And look how much larger these particle sizes are looking at comparing those two diagrams. So the particle size is going to be larger. One of the relevant examples here for the biological example is going to be blood. Do we get the formation of sediment in this example? Yes, we do. The suspension, this is a heterogeneous mixture. And do solid particles settle out? Yes. You get sediment at the bottom. And is the pathway going to change whenever you shine the light through it? Yes. The pathway is going to change. So if we separate these two components here, can someone tell me what is this top part of the blood? So this is the plasma. And then here at the bottom, these are the formed elements. We've got red blood cells, white blood cells, going to be found here within the formed elements of blood. How they're able to separate this is they put it in a centrifuge, and then it spins it around. It separates based off of its density. All the components in blood, we talked about water, all the different solutes, all the salts. Is hemoglobin a protein? Hemoglobin is the carrier that transports oxygen. All of those proteins, it's going to have a lot of mass, so it's going to be found here at the bottom because it separates based off of its mass. Non-biological example here of a suspension. This is going to be a mixture of sand and water, which is what we looked at earlier. A biological example here, we talked about the blood. So living blood cells within the fluid portion or the blood plasma. Distinguishing between a compound and a mixture. So the primary difference between these two, between mixtures and compounds, no chemical bonding occurs between the components of the mixture. When we mix them, their properties are not going to be changed. They're only mixed physically. Means of separation between the two, mixtures versus compounds. So mixtures, the components are going to be separated by physical means. Using that centrifuge, that's a physical means in order to separate those two components, as opposed to separating these compounds, you have to actually break bonds. So that's by chemical means.